Welcome to the Chemistry, Biology and Math Revision Hub. Today we are doing the Pearson Edexcel Level 3 GCE Biology B Paper 1 for June 2022, and this is the Part 3 video. I'll put the links to the Part 1 and Part 2 videos below in the description box. Let's begin. Question 8 says, hypoxia is an inadequate supply of oxygen to tissues and cells that restrict their function. The normal partial pressures of oxygen vary from tissue to tissue. The table shows the normal partial pressure of oxygen into tissues. So we have tissue and the partial pressure of oxygen. One is the pulmonary arterial blood, which has that partial pressure. The other arterial blood that have that partial pressure. So they say calculate the ratio of partial pressures of oxygen in these two tissues and give your answer to one decimal place. The ratio should be pulmonary arterial to other arterial 5.3 to 13.3, which is the same as 1 to 2.5. So you could say 1 to 2.5, or you could say 0.4 to 1 if you had divided the other way around. So my answer was 1 to 2.5. And down here they say explain why the partial pressures of oxygen in these two tissues are different. When we look at the information that is given, we can see in pulmonary arterial blood, this is carrying the oxygenated blood, that's why the partial pressure of oxygen is quite low, but other arterial blood is going to have a higher partial pressure of oxygen because it's carrying oxygenated blood to the rest of the body. So here I said, pulmonary arterial blood has a lower partial pressure of oxygen because the pulmonary artery is carrying the oxygenated blood from the heart to the lungs, and other arterial blood has a higher partial pressure or have a higher partial pressure of oxygen because those arteries carry oxygenated blood towards tissues or organs of the body. Moving on. Here they say the body responds to hypoxia by releasing hypoxia-inducible transcription factors, HIF. Investigations have shown that one effect of HIF is an increase in the rate of glycolysis in the affected cells. Explain how HIF could result in the increase in the rate of glycolysis. So any factor that is going to induce cells to react in a specific way should be able to either bind onto receptors or to be able to enter into the cell. So here I said, the produced HIF could cause increased gene expression, meaning acting as a transcription factor. It can bind to a promoter region and initiate transcription, and this can result in production of proteins or enzymes involved in glycolysis. And down here they say, explain why cells need to respond to hypoxia with an increase in the rate of glycolysis. Now we know glycolysis leads to formation of pyruvate, and this pyruvate could be taken through anaerobic respiration if there is insufficient oxygen. So the rate of glycolysis is going to increase in order to provide ATP through anaerobic pathways in order for the body to function. So I said, during hypoxic conditions, there is insufficient oxygen, and the electron transport chain will not operate because oxygen is required as the final electron acceptor during aerobic respiration. ATP production will be reduced, so substrate level phosphorylation will occur to produce ATP through glycolysis under anaerobic conditions. Here they say the graph shows the changes in level of two HIFs, HIF1 and HIF2, before and during hypoxia. Before hypoxia, the levels of HIFs are both low, although HIF2 is higher than HIF1. However, during hypoxia, we see they both begin to increase. However, HIF1 reaches a maximum and decreases, but HIF2 seems to decrease quite slowly or actually kind of maintained a little bit constant. So here they said compare and contrast the changes in the levels of HIF1 and HIF2 during hypoxia. I said both HIF1 and HIF2 increase during hypoxia, and the levels of HIF1 increase and then fall, while the levels of HIF2 increase and remain high. Down here they say explain the changes in levels of HIF1 and HIF2 during hypoxia. This is explaining why they do increase. So I said HIF1 and HIF2 cause transcription of different genes, leading to synthesis of different proteins, and then the products resulting from presence of HIF1 and HIF2 are needed during the early stages of hypoxia. Products resulting from presence of HIF2 are needed for long during hypoxia than those from HIF1. So this brings us to the end of question 8. Let's continue to question 9. Question 9. Plants and animals are adapted for gas exchange. Which row of the table describes gas exchange in the root of a plant? Now, net movement of carbon dioxide is going to be out only. There is no need for CO2 in the roots because they do not carry out photosynthesis. And then net movement of oxygen is going to be in only. And again, because there will be no photosynthesis, so there is no release of oxygen. So the answer should be a B. The next part they say, which is the gas exchange surface in the stems of a woody plant? The answer here should be A, which is the lenticule. Moving on. Here they say, spiracles are small opening in the exoskeleton of insects that allow air to enter the respiratory system. Water can evaporate out of the spiracles when they are open. The insect can close the spiracles to reduce water loss. In an investigation, the water loss from insects in air with different humidities was measured. The insects were kept in air with 80% humidity and then moved into air with a lower humidity, and the water loss was then measured. The investigation was repeated in air in carbon dioxide to keep the spiracles open. The table shows the results of this investigation. So here we have the percentage humidity as it increases, and then the water loss from insects. We can see here insects in air and insects in air high in carbon dioxide. The first question says, state how the water loss could have been measured in this investigation. So here we could do that by measuring the mass of the insects before and after the experiment. If there is a decrease, the mass of the insects will go down. So I said by weighing the insects to obtain the mass before and after the investigation and then calculating the difference in mass. In the next part they say, calculate the percentage increase in water loss from the insects kept in air at 0% humidity compared to those kept at 80% humidity and give your answer to 2 decimal places. Those kept in 0% humidity, you can see 0.1, and those kept in 80% humidity is 0.07. So the difference is 0.1 minus 0.07 divided by 0.07 times 100. So I got 42.8571. But they require us to write the answer in 2 decimal places, so it's going to be 42.86%, which is the answer here. Moving on. Here they say, explain why the insects were kept in air with 80% humidity at the start of this investigation. So I said, to prevent water loss through the spiracles before the experiment, the 80% humidity was used as a standard for comparison, and this allows to study how lower humidities affect water loss. Next they say, explain the results of this investigation. We can see based on the table, as percentage humidity increases, we see there is a decrease in water loss for both insects kept in air and those in air with higher carbon dioxide concentration. So here I said, as percentage humidity increases, water loss decreases. There is greater water loss in air with higher carbon dioxide because presence of carbon dioxide leads to a higher ventilation rate, and this maintains the spiracles open. Remember, when there is a higher concentration of CO2, insects want to take in more oxygen, so the spiracles are going to be kept open for longer, and therefore more water will be lost. Moving on. Part C says, the gas exchange surface of fish are the gills. The diagram shows part of the structure of a gill. So we can see this is the structure. Down here they say, fish can be divided into 2 groups, species of fish that are active and species of fish that are inactive. The table shows some information about some species of fish and their gills. So we can see a lot of information here that we are going to refer to in the next question. Moving on. Here they say, lamellae are present on both sides of the gill filament. The mean length of a gill filament is 25 millimeters. They want you to calculate the total number of lamellae on the gill filament of Thanos and express the answer in standard form. Before I go through my answer, I want us to look at this explanation. For Thanos, we can see there are 24 lamellae on each side of the filament. So remember, they also told us the length is 25. So 24 on each side times 25 gives us 600. So in my answer here, I said if these are 600, the total is going to be 1,200, because if these are 600 and the other side 600, we have 1,200. So I say the total number of lamellae in each filament are 2 times 600, which is 1,200. So the total lamellae in the filaments are going to be, if we go back here again for that, this one here, it's going to be that. These are the total number of gills, 6,480. So we can say 6,480 times 1,200, which gives us that. In writing this in standard form, it's 7.8 times 10 power 6, and that is my answer. Please revisit this explanation for more details. Moving on. Here they say, analyze the data to explain the relationship between the activity of these fish and the structure of their gills. From here we can see these are active species and these are inactive species. We can see the active species actually have a higher mass. The total number of gills are more. And then we can see the number of lamellae on each filament are also high, except this one here among the inactive fish. The surface area is also high in these compared to that. So I said, generally there are more gill filaments among active fish than in inactive fish, except for the 268-gram tinker. And generally, active fish have more lamellae on each filament than inactive fish, but there is no correlation between the number of lamellae and the mass of the fish. And again, you can see that from the table. I say Trachurus is the fish with the most number of lamellae, there were 39, while Ictalurus has the least number of lamellae, there were 10. Also, generally, active fish have a greater surface area of gills than inactive fish of the same mass, though there is no established correlation between the mass and the surface area. About the diffusion distance, there is not enough data for both active and inactive fish. However, based on the provided data, active fish seem to have a smaller diffusion distance. And finally, the greater the number of filaments, the lamellae, surface area, and lower diffusion distance allow for efficient gas exchange, so oxygen and nutrients are available for aerobic respiration to provide ATP needed for muscle contraction. This one here is about the processes, the details of the processes that occur. For every 6-mark question that you're required to answer, ensure that you can write the details of the physiological process. Whichever physiological process is going on, try to explain the details. In this case, it was about diffusion, gases, and so on. So we have to talk about the physiology behind that and how this diffusion of gases is going to be helpful to the organisms that are carrying out gases exchange. So this brings us to the end of this video, as well as to the end of the whole paper. Thank you for being with us. Do not forget to subscribe. See you in the next video. Bye-bye.